So that's in terms of true CME. I'm actually going to spend a lot of time talking about recent data with respect to the clearance systems of the brain across many labs, across rodent studies and human studies. And then if I have time for it, I'm going to talk about some of the things that we're doing at Wisconsin to attempt to manipulate and improve clearance as well for a variety of diseases and pathological dysfunction. So a lot of disclosures. One of the things, if you haven't met me before, I do a lot in terms of industry and company work. If I can ever help you, if you know nothing about this talk or take nothing from it, I'm here to help you. I'm a professor within the Department of Neurosurgery, and I'm an engineer. And I've taken a lot of device ideas from concept to clinic, all the way through to FDA approvals. But more importantly, two of these, I'm going to be talking about cranial nerve stimulation. I have the scientific advisory board of LivaNova, which implants vagus nerve stimulators, and then I am a co-founder of a company called NeuroWorks, which is attempting to leverage some of our ideas for cranial nerve stimulation to facilitate clearance of the brain in diseases and disorders. So you all should at least know what the traditional lymphatic system is. And I'm going to point to a couple of things in terms of common lymphatic function. We all know, if we exercise, we get lactose in the extracellular space. We need to clear these molecules associated with it, and we have the traditional lymphatic system, which is largely responsible for it. And we have these partial pressures at capillary bed that kind of create movement. But an important part of the traditional lymphatic system is we have these endothelial cells that pressure causes to move, that allows molecules to kind of move through the space. Now, it's interesting if you start thinking about brain clearance and why this wouldn't be an appropriate system for brain clearance, because we need to keep immune privilege. There's a reason we have the blood-brain barrier. So for a very long time, we had no idea, to some extent, how the brain clears metabolic waste. We still get lots of molecules in the extracellular space that we need to get out of the brain for a variety of reasons. The other thing to point out is that the traditional lymphatic system, once you kind of get in through these capillary beds, is thought of as a duct-based system. This means it's you have valves that create directionality of the lymphatic flow. Now, that's probably what you were taught. I don't know if you've also been taught that there's actually neural innervation, especially at the lymph nodes of the lymphatic system, that actually changes oscillatory function of the lymph nodes. So what I was taught originally, I think what a lot of you were probably taught, is that it's a completely passive system. It's actually not, and there's actually axons that are non-canonical branches that innervate the cervical lymph nodes, and coming off the sympathetic trunk that provides sympathetic and parasympathetic innervation of the lymph node that changes its oscillatory function. And they're pretty much impossible to see surgically because they're diffuse axons as well. So why do we know about the lymph nodes and the cervical lymphatics? And the truth of the matter is, in dissection, really obvious to see. You see these large lymph node structures all over the body. So Hippocrates actually had identified lymph nodes. And then once you see these large structures, you're kind of, okay, what are they connected to, and all that sort of stuff. So the brain does not have these visible lymph nodes, often attributed to this immune privilege idea. So the question is, how does the immune privileged brain clear interstitial metabolic waste of molecules? Note this is also important for anesthesia. I've talked to a lot of anesthesiologists, and they think about getting things into the brain, but not about how it leaves the brain and the dwell time. And this becomes potentially really important for things like postoperative delirium, where anesthetic, stress hormones all kind of go together and may create inflammation over time as well. So a seminal paper in 2012 by Jeffrey Eiliff and Megan Nedegaard really kind of started this resurgence into looking into how the brain clears things. And they did a really cool study. I'm going to show you some complicated figures. I'm going to point you to just a couple of them, so you'll know all of them. And I'm just going to give key points to a lot of seminal papers that have come out since 2012. But in this paper in 2012, they said, okay, let's just inject a dye in the cisterna magna, and let's see where it goes. And the cool thing now with rodents is you can do 2-photon microscopy, and you can actually visualize the dye kind of going up, then going down what they saw, which is the paravascular spaces. So after they injected a dye in the cisterna magna, they started to see the dye go down the paravascular spaces of the MCA. Then as you get the brain parenchyma, you see later on it starts getting out, filling the brain parenchyma. So, you zoom in and you see these perivascular spaces around the major arteries and around the descending arterioles. So then they started asking a question. They noticed that these perivascular spaces were defined by astrocytes. They're actually created by these astrocytes. So they asked the question of how is CSF kind of getting down these perivascular spaces and into the parenchyma? So they did staining to look for AQP4 channels are a water channel that allow pretty much water and very small molecules to get through. And the astrocytic end feet had these both tight junctions in which fluid could get through, but also had these AQP4 channels. So they did a couple of really cool things in terms of looking at wild type mice and AQP4 knockout mice to see if CSF would go from the cisterna magna down the perivascular spaces and in the parenchyma if I knocked out these AQP4 channels versus I hadn't. And they looked at it in a variety of ways. So they would inject interstriate, which is this idea that, okay, I'm now putting a molecule in a radiolabeled molecule kind of in the brain parenchyma, and does it get out of the brain? And so what they saw is in the wild type animals that have intact AQP4 channels, if they inject a radiolabeled tracer right into the parenchyma, it gets out of the brain. But if they have the AQP4 knockouts, it does not get out of the brain as well. This led them to postulate what's called the lymphatic system, because astrocytes are glial cells, which is an analog to the traditional lymphatic system. And in this model, what you have is the arterial system is as it pulsates, as you have slow vasomotion, it moves CSF in the perivascular space through these tight feet and AQP4 channels into the parenchyma. And instead of just molecules being cleared by diffusion, it creates an advective gradient. So they looked at this also in terms of, you want to do it this two ways, right? You inject in the parenchyma and see does it get out of the brain, but then you inject in the CSF and you see does it get into the brain, wild type versus AQP4 knockouts. What they found out is that in the wild type, inject in the cisterna magna, it gets into the brain. If you knock out these AQP4 channels, it doesn't get into the brain nearly as well. So the astrocytic end feet don't serve as well as these AQP4 channels. Now, there is a thought that these AQP4 channels are actually preferentially open and polarized towards the perivascular space during slow wave sleep, which is why I enjoy giving this conversation to a bunch of neurosurgeons and neurosurgery residents who have been sleep deprived for most of their career. So, sleep. They did a follow-up study. Sleep deprives metabolite clearance from the adult brain. So, all they did is a simple study, same study largely, but then they're saying sleep versus awake. And then they also did awake versus ketamine-induced anesthesia, because ketamine mimics slow wave sleep. And what you can see here is that awake, the tracer doesn't get down these descending arterioles into the parenchyma, and that asleep, there's much deeper penetration over a short period of time. And similarly, ketamine versus awake, same sort of difference. So this led to them thinking of a couple of things that is driving this during slow wave sleep. So it's not just that these AQP4 channels are open, it's that you get these slower vasomotion waves during slow wave sleep that drive CSF in the parenchyma, but also it's important to have quiescent neural activity. And the reason that is because in the interstitial space, cells that are more active actually swell. If they are less active, they actually expunge some fluid content. And that means if they expunge the fluid content, the interstitial pathways have less resistance to them as well. So there's multiple parts of this system in terms of that you might want to manipulate to increase this advective gradient that clears from the brain. So
I'm getting confused. The perivascular fluid, you can pump it into the brain.
Right. How does it get out?
How does it get out? Because the
I'll get that
molecules are there.
There's several routes in which it gets out that they found now. And I'm not going to go that into great detail. There's also a little bit of controversy. Sometimes it's actually out just through the blood-brain barrier and active transport, and you're just increasing interaction with sites of active transport. There's also egress out the perivascular spaces of the veins. There's also egress out the cribriform plate that drains in the cervical lymphatics. And then finally, the major cranial nerves all have paraneural fluid that there's egress out of. In fact, to some extent, that if you inject a dye into the brain, into the CSF, that you will actually get it in the paraneural spaces, paraneural fluid in the peripheral nerves, which is an interesting thing as an assay. You actually get appreciable CSF from the brain to the spinal cord, but also out to sciatic nerve, for instance.
So you're arguing that the molecules are cleared by a river which is pumped through the brain, comes out through various channels.
I'll say that is how many people take it. I put one step further because there are gates in between that are permeable to certain molecules and active and passive transport. So to me, that river just increases interaction with these sites. That then active transport might get over these various barriers. And then as the concentration gradient changes, you're also going to have an osmotic gradient that will drive additional kind of fluid movement as well. That's my hypothesis. I don't see it much in literature. But most importantly, if this video plays, this is injecting fluorescent dextran. This is the perivascular spaces. And the important thing to note is that every arterial and respiratory motion, you see these little molecules move forward. Now, the traditional lymphatic hypothesis is that the CSF moves, that the slow vasomotion is driving it like a peristaltic pump and driving the CSF out these AQP4 channels. There's also some evidence that it might just serve to, there might be reverse directional flow depending on how they're pulsing, and that it might just agitate it as well to create more, which is called the mixing hypothesis. So there's the lymphatic hypothesis, what's called the IPAD hypothesis, which is CSF moving in the opposite direction this is showing. And then there's this mixing hypothesis, which is moving molecules, and they're going to interact with these sites actively. So then it becomes, where do these drain to? One of the places that these drain to is what are called the meningeal lymphatics or the dural lymphatics. Now, the interesting thing is these had been identified as early as 1787, and everybody ignored it and said there's not a lymphatic system in the brain. This is more the passive duct-based system that everybody knows about. So Jonathan Kipnis's group did some great work looking along, at first in rodents, along the dural sinuses, and found that they could find lymphatic vessels co-digitated with dural sinuses that have more of this traditional duct-based structure. And now, as you think about lymphatic, there's this idea that during slow wave sleep, the lymphatic system operates and drains to the meningeal lymphatics, but then the meningeal lymphatics drain roughly to the cervical lymph nodes more through awake behavior and relative motion and moving through this duct-based systems. So evidence, this is all rodent work at this point, right? So the question is this just a rodent phenomenon, or what's the evidence that they're humans? So first of all, not directly measuring it, but Laura Lewis's group did a great study where they did, they looked at EEG to look at slow wave sleep. They looked at changes in fMRI bold signal, and then they looked at CSF in the ventricles, which they could resolve. And they found a very tight couple between slow wave sleep, this changes in this deactivation of bold, and then this an increase in CSF movement in the ventricles. I'm not going to have time to get all into this, but in the lower right-hand corner, you'll see that in specific, the negative change in bold correlated amazingly well with the movement of CSF and these oscillations that you see. So then people started just doing in large animal models, injecting gadolinium and other contrast agents intrathecally, and they started showing, similar to the rodent studies, these molecules moving kind of up and down the perivascular spaces as well, and identified these meningeal lymphatics. So the question becomes, why did we not know this before? And so this is a rodent study, 2-photon, that shows you the outline of the perivascular space around the artery and the vein. And the truth of the matter, these are incredibly thin membranes that when you do histology, if you're not really smart, they collapse. So you just don't see them. So more recently, this was published in the last year, Make and Nedergaard did a really interesting study connecting not just slow wave sleep, but norepinephrine behavior. So locus coeruleus is really supplies most of the norepinephrine to cortex, right? and that locus coeruleus oscillations, which is connected to sleep, actually creates blood flow changes that then drive CSF movement. So the blue is really CSF movement. This is blood flow changes. This is EEG and EMG. But then what's really cool that you can do in rodents is you can do optogenetics. So they actually optogenetically activated LC and then looked at change in CSF movement at different, so they're going to activate norepinephrine, right? And what they see is you see these spikes in norepinephrine. So they're measuring norepinephrine noradrenergic increases in their behavior via a GCAMP, which is a fluorescent indicator of neural activity. They're showing that, hey, we're seeing increased neural activity. Then we're seeing the CSF tracer. We're seeing increases in the CSF tracer that actually correlate. But then they can give a norepinephrine antagonist in this case. So they give a norepinephrine antagonist, and then the function changes dramatically, connecting it to LC's norepinephrine influence on cortex in this case. Something that they overlook in this paper that's important for what I'm going to discuss as well, if we have time for it, is LC provides norepinephrine to cortex. But when you get to the large major vessels of the brain, most of the sympathetic and parasympathetic innervation is actually from the trigeminal sensory nucleus and the sphenopalatine ganglia, not locus coeruleus. So they kind of missed that aspect of all of this as well in their paper. They don't discuss it. So then it becomes in humans. So if slow wave sleep is supposed to be important, can we do lumbar catheters, measure CSF changes during sleep and sleep-deprived people, and measure blood plasma and see whether we get changes in amyloid beta, phosphorylated and unphosphorylated tau. So this was done by Brandon Lucy's group, and what you're going to see is during the circadian cycle where they're supposed to be sleeping, what happens in sleep-deprived individuals, you'll see all of these misfolded proteins, often or proteins that can be misfolded that are correlated with Alzheimer's disease and some of the Parkinson's disease and some of the outcomes of TBI, they go up in the CSF. And if you get people who are sleeping normally, they don't go up in the CSF, but they go up in the blood. So this is really interesting just in terms of how we even look at our biomarkers. We're pulling stuff from the blood and saying, do we have elevated levels of GFAP, neurofilament light, tau? But to some extent, getting out to the blood is getting rid of it from the brain. We might not be thinking of it in the same way that we should be, that we need to get this out to the blood. And then at some point it would spike up, but then it would go down over time as well. So now you can imagine the lymphatic system, if you buy this as a hypothesis, there's lots of things that would impact its function. First of all, just aging. As your arteries become more stiffer and things like that, you're going to change that pulsatility. If you get a subarachnoid hemorrhage, you're going to damage these perivascular spaces too. And you're going to get blockage of the system. In Alzheimer's disease, you get amyloid plaques that also create amyloid angiopathies that stiffen the arteries as well. So it creates these unique slippery slope systems. There's even evidence suggesting that every time we put an implant in the brain, you get tau, you sever these systems, and over time, these sticky proteins create seed points for more sticky proteins to aggregate. You have a long time course before you start getting inflammation, cellular degeneration. You start the slippery slope cycle of more sticky proteins as well. So this is a rodent study that's incredibly cool that also just came out from the Nedegard group, and one of our collaborators, Jeff Titoff, is on this, where they looked at if we do TBI, how does drainage out to the cervical lymphatics change from this lymphatic system? And so they're looking at the cervical lymph nodes, and they're doing a couple of perturbations. First, they're showing that you have GFAP positive cells and NUN positive cells that get to the cervical lymphatics. They're kind of the cellular debris, these damage-associated molecular patterns that create triggers for inflammation, and that in normal function, some are draining because you have natural loss of some of these things as well, cervical lymphatics. And TBI is compromised because you have this norepinephrine spike that actually compromises lymphatic function. And then if they get a norepinephrine antagonist to restore lymphatic flow, these damage-associated molecular patterns start to drain the cervical lymphatics. And you see a notable increase in the amount of cells, these damaged fragments in the cervical lymphatics as well.
There's a lot of interesting implications for all of the biomarkers that we're looking at. right? We're using those as surrogates of brain injury, but you have to get it out of the brain in order to study it in the blood. And if the mechanisms for getting it out of the brain are impaired, then your damage is going to look like a lot less, even though you're still built up.
Well, and that's the thing. If you're doing a treatment, say you're doing a norepinephrine antagonist, you should see more getting out to the blood. And normally we take that as a problem, but really we need to get it out, and then it should go down over time, right? So it's a very different way of looking at it. So then when we get to humans, it's hard to visualize CSF and perivascular spaces. They're very small, so a lot of times people look at the ventricles. There's been a technique that's commonly used that has some problems with it called DTI ALPS. DTI, as probably everyone knows, you can do tractography with it. You're essentially resonating water molecules, and you're looking where they move. Well, they want to take advantage of a structural feature to say, okay, I want to look at movement of water along the perivascular spaces, not along fiber tracts and things like that as well. So they look at a very specific location in the brain where the medullary veins run in the DX direction. The projection and the association fibers run in the DY and the DZ direction. Then to decouple from blood flow, they're actually using a B field that will highlight slow movement versus faster movement. And so there's been, honestly, hundreds of studies at this point showing in addition, and many things that should compromise lymphatic function, you see a decrease in what's called the DTI ALPS index, which is a ratio of the DX direction, which should be CSF flow, to the DY and the DZ direction. And you see with increasing severities of TBI, reduction of this index. It also happens to be the Parkinson's disease and Alzheimer's disease. They've shown this stroke, et cetera, as well. Now, it's important to note, though, this isn't the influx tract for the lymphatic system. This is one of the outflow tracts that's being compromised along the medullary vein. So then I became very interested. Most of you know, have a background in stimulating the sympathetic parasympathetic nervous system. So when I was an NIH program director, these studies started coming out, and I went, well, I went to humans and took a product to stimulate the sympathetic parasympathetic nervous system. Does that actually have an effect on the lymphatic system as well? So I'm going to skip through this really quickly, but if you stimulate cranial nerves, whether it is the carotid sinus nerve, the vagus nerve, or even the trigeminal nerve, you get input to NTS, which then projects to the caudal and rostroventrolateral medulla, which are essentially the sympathetic parasympathetic outflow to the rest of the body. In addition, the trigeminal sensory nucleus projects to the large major vessels and controls the smooth muscle as well. There's also a recent paper showing that the trigeminal sensory nucleus is fenestrated and has sensors for molecules in the CSF at the trigeminal sensory nucleus. So there may be a feedback loop through trigeminal sensory nucleus as well. So we've shown in canines and in humans, I'm not going to get into this too much, that we can change blood pressure, dilate vessels by stimulating, in this case, the baroreflex in canines as well. So this is just a longitudinal study in canines showing the blood pressure change over a week. This is showing acute blood pressure changes, which is surrogate for dilating the vessels. So there's a lot in terms of vagus nerve stimulation to treat epilepsy. And one of the things that struck me is we're not very good with the mechanism for vagus nerve stimulation. And maybe the way it's working is causing CSF to increase in parenchyma, having a number of different things. It's going to have a dilutive function for transmitters, but it will also reduce non-synaptic coupling. If I increase the distance between cells, maybe it's been shown certainly in cell cultures and organoids, if you induce a seizure, if I just do isotonic saline, I'll abort the seizure as well. So we did vagus nerve stimulation in a variety of different fashions, and we did the same thing that Edgar did, inject a dye in the cisterna magna. And what we saw is a statistically significant of CSF or the dye into the brain parenchyma versus controls and shams. But importantly, we had to do temporally pattern stimulation. We had, in our view, we're creating slow vasomotion similar to cardiac and respiratory cycle, but slower because we don't want to interact with it. And if we stimulate it continuously, it didn't make a difference. I've done a lot of blood flow studies, and we have a lot of homeostatic mechanisms. Brain likes to keep constant perfusion. So if you stimulate the cranial nerves continuously, it adapts pretty quickly. You'll get a transient blood flow change, but then it goes back to baseline. That you have to do this pulsatile activation, allow it to go back to normal, and pulsatile to actually drive CSF. So then this becomes possible. If we can do this becomes a treatment for a variety of diseases and disorders. Keep looking at my time, but I wanted you all to be aware of this study because I don't think it's resonated as much as it probably should have. It's Nature Medicine, but this is a study of a group of subjects were given human cadaver pituitary-derived growth hormone in the UK between 1959 and 1985. What they found was that many of these samples that they were given were contaminated. Specifically, they had amyloid beta prions. So they demonstrated that the ones that had amyloid beta and tau contamination, that if they put it in mice, that they went on to have dementia symptoms. But then they found some subjects that got the contaminated and they looked at them, and 5 of them went on to early dementia. So they got this in their teens. Onset from 38 to 49 years old. So you're talking in terms of the prevalence of the population. This seems very likely to be causal, that these amyloid beta prions that they got from the pituitary growth 15 years later, roughly, was enough to create the seed sticky proteins that then created these aggregates that then caused inflammation. So this is the first evidence of human to mouse transfer via amyloid beta and tau of Alzheimer's disease, and then human to human as well. Obviously, it's unlikely for this to happen in normal behavior. This is a rare circumstance that it was transferred human to human putatively. So because of the slippery slope problem for a lot of these diseases and disorders, and even in TBI, where we want to get rid of the damage-associated molecular patterns, prevent them from triggering edema and inflammation, creating secondary insult, we probably don't want to do an invasive vagus nerve implant for a lot of these subjects. We want to intervene as early as possible. So we've done a lot of animal and human work looking at different ways to stimulate the periphery and reliably engage. Yes, that can change sympathetic. Really, I'm gonna run out of time. But the big problem is a lot of these studies, clinical studies, have run into side effects that prevent you from actually activating these pathways. When you try to electrically activate these nerves, you activate other nerves that activate the neck muscles, the muscles of the forehead. They also activate nociceptors in the skin. Problematic, especially for non-invasive. We can defibrillate the heart from the surface of the skin. We cannot do it at a comfortable level to facilitate clearance during sleep. Surprisingly, a defibrillator is probably going to wake someone up, stop their heart. Gonna skip this, but so there's a lot of people saying they're stimulating the cervical vagus, which is 2 to 4 centimeters, using non-invasive stimulators. We've done a lot of work. You're not. You're just in all these cases, you're typically stimulating superficial nerve trunks right above the muscle, and then you're activating the muscle, and then patients tell you to stop. This also is, so we're doing a lot of anatomy work across human cadavers just to show how complicated the human anatomy is. This is a micro CT. That was, if you're stimulating the cervical vagus from here, you have to activate so many different nerves, like the phrenic, you'd be activating the sympathetic trunk. And for whatever reason, these people tell you they can magically stimulate just the cervical vagus and maybe the superficial neck muscle, but they're not creating diaphragmatic contractions. So we've done a lot of animal studies. We don't believe you can stimulate the cervical vagus. You can get certain spinal nerves that might modulate NPS as well. We also looked at stent-based systems to run it up the IJV and stimulate the vagus. And again, you can do it, but you can't do it at levels that are not going to stimulate a lot of the surrounding tissue and create side effects as well. In fact, limiting side effects. You don't activate the small diameter fibers going to the brain before you activate these side effects in patients. Yeah.
So I understand why you chose vagus nerve because it's a common site for stimulation, but
we're done with Vegas.
less common, but still totally feasible are trigeminal sites and stellate ganglion sites.
Let's roll. So we did a lot of modeling to show what are these superficial nerves and skin sensory receptors that prevent you from doing it, and can you actually activate it? You just jumped to punchline. We did the auricular vagus as well, and we went to the trigeminal. So the trigeminal, most of the companies that are stimulating it believe they're stimulating as a component under the muscle. We do not believe that's the case. You're actually stimulating the supraorbital and supratrochlear, kind of like when you inject lidocaine, just right above the muscle as well. So trigeminal nerve has been shown across animal models and humans to reliably change blood flow through a multitude of mechanisms. Not going to have time to get into that, but importantly, as Don is implying, it's superficial. It's right above the muscle, and I can stimulate it without activating the A-delta nociceptors in the skin. And what's really cool is because it innervates most of the forehead, scalp, the crown, if I'm not just activating the skin under the electrode, but activating this nerve trunk, I will get a clear referred sensation that goes into my head. It's an electrical tingle / massage feeling. If I turn it up, yes, I'm a stupid enough idiot to do this. I can get A-delta nociceptors through it, and I will get a referred burning sensation that feels like some cautery or bee stings in my head. The other important thing about it is in terms of off-target nerves, the facial nerves are motor muscle activation that we want to avoid as well, but they actually run in orthogonally. So with really intelligent electrode design, instead of these huge patches that people are using, we can actually avoid the superficial anatomy that creates the side effect and stimulate what we want. The other nice thing about it is, in addition to invasive MRI-based measures, we can use what's called F-NIRS on the headband to get a readout of blood flow changes in prefrontal cortex. Trigeminal nerve stimulation stimulates trigeminal sensory nucleus, NTS, but also has projectivity to VTA, which projects to prefrontal cortex. So you'd expect to see prefrontal cortex changes in blood flow as well. And you can have a headband system that's smart, that has this F-NIRS, non-invasive, to do a closed loop system.
I would take it even a step further and say, surgically, we have pretty safe access to the trigeminal tissue to stimulate there to get maximal response.
Talking to a lot of people about doing proper studies for that purpose as well. And we'd love to continue that conversation with you. Go through animal model studies. So this is stimulating, in this case, other nerves. This is the buccal branch of the facial nerve. This is doing a Doppler flow ultrasound through craniotomy, but this is intended to show you in large animal models that you get blood flow changes. That's a blood flow change. But more importantly, the time course of the blood flow change hits our habituation hypothesis, that it peaks into a 30-second pulse train pretty quickly, and it goes back to baseline before you're done as well. So that traditional trigeminal nerve stimulators that stimulate continuously or aren't appropriately temporally patterned may hurt the lymphatic system or may not be messing with it at all. So then we did NHP studies and showed we could stimulate the V3 branch of the trigeminal, the mental nerve, that when we stimulate, we can create bradycardia or tachycardia. This is positive bold in the NHP in the brainstem, where you expect the trigeminal sensory nucleus to be, and then you actually see broad inhibition across the brain as well. So cranial nerve stimulation activates inhibitory neurons, it activates GABA. So you'll actually see the kind of deactivation as well. So then we used DTI in a very specialized way to look at movement of water molecules along the paravascular space. So this is in the NHP, this is the paravascular space around the MCA, and these lifts show directionality of movement. You can see they're kind of moving around the paravascular space, and then they kind of go orthogonally through these AQP4 channels out to the parenchyma. And then when we stimulate, they organize, and you see more flow along the paravascular space, and you see more organized flow out these AQP4 channels into the parenchyma. And this is what it looks like in rodents under 2 photon as well, just to show you that this is exactly what you'd expect in NHPs, that this method is working data vision-wise. So independently, several labs have now looked. We published the temporal patterning study. They've done the temporal patterning of the trigeminal stim and found recovery in TBI, including increases in cerebral blood flow, reduction in brain edema, recovery of multiple mental tasks associated with TBI in the rodent model as well. So finally, I got like 5 minutes left. I'm going to quickly show you some human data as well. So we started healthy human subjects. We've also gotten, we're enrolling right now with the Dept of Neurosurgery, TBI as well, and looking at mechanistic engagement. Can we stimulate blood flow changes broadly within the brain and CSF? So done a lot of cadaver studies to optimize this electrode design to get the this is work done by Erin Szyminski and Denny actually just got published as a first author paper for her as well in Journal of Neural Engineering. So this is no longer archived. We've done a lot of modeling of these different electric designs viably across a cohort, not just one person given the variance. Activate, get a referred sensation, not get this off-target stuff. Skip. So this is these healthy subjects saying what referred sensation they get. And what we found is that with a suboptimal MRI compatible design that we're trying to place appropriately, not our final system, which we believe we can even improve upon this, we're able to get a comfortable referred sensation that they feel is comfortable for use during sleep. We did a bunch of MRI measures, several of them fell asleep as well. So then we do several measures that look at both CSF influx into the brain, brain blood flow, and then fMRI to say, are we just activating a small portion of sensory cortex, or trigeminal going into your brainstem through multiple midbrain nuclei should broadly change the brain. So this is 6 subjects. What you're going to see is positive activation in brainstem and in sensory thalamus and other midbrain structures. And you'll see this mix of positive and negative fold. You'll also see it's pretty bilateral, that we're not just getting one side of the brain because we're stimulating both sides as well. This is comparing the FNIRS response to the fMRI response. The fMRI response, if you look at it closely, before the stimulation period and goes back like we expected. Again, suggesting that if we stimulate it constantly, it would habituate, create the slow vasomotion. And then if you get no referred sensation, the FNIRS shows essentially what's called a myorhythm, a slow oscillatory signal. You get the referred sensation, you see a large blood flow change putatively in motor cortex that kind of matches the fMRI in terms of the time course for the same individual as well. So this connects our non-invasive measure to a more known measure as well. And then finally, does it create CSF movement along the perivascular spaces? In short, this is a TT-weighted image. This is the MCA. This is the perivascular space. This is similar to what you just saw in the NHP. This is unstimulated. Red is the R to L direction here. And you see when we stimulate, we see increase in movement along, or increase along the perivascular space direction. And it's statistically significant already in a relatively small group. So we see, and importantly, because we wanted to assess whether this was an accommodation effect, being in the MRI or and changing their sympathetic tone, their baseline of this measure goes up stimulated, goes back down once they stop being stimulated. So it does not appear to be an accommodation. All right, that was an incredible amount in a short period of time. There's been so many people within neurosurgery, radiology, and metaphysics that are part of this. You can't do this without cross-departmental collaboration. Also would like to thank funders, especially Fund of Neuroscience, also created a small grant to get this unique data as well. A couple minutes for questions.
If I hadn't had as much fun doing grand rounds in a long time, because I've always been fascinated by the pulsatile secondary effects of heartbeat and pump, and the second pump. It's like keeping the vacuum of a internal combustion engine that drives electricity in a car. And
Also, how we're changing that system.
Yeah. This is beautiful work. I think I want people to just sort of stop and think, because that's what you're helping us do. Question is tying this to disease, and the big thing we've talked about is measuring these waste products. You're showing their highway. We've got to figure out ways to measure them in situ.
And this is where Patrick has actually done an incredible guide for what biomarkers for TBI and really what's the SNR of them. Now, the Lucy study suggests that I can do a relatively small amount of subjects and at least see changes in phosphorylated and unphosphorylated tau. If we get effects, maybe a third of that, we should be. So I should say, in terms of our measurements, there's a study that looked at CSF movement in sleep versus awake in healthy human subjects, and we're seem to be getting about half of that in terms of our DTI ALPS index change as well. But the question is that enough? How much blood biomarkers does that change? The other thing is, we've been talking about it in terms of when they do these biomarker studies, they're not controlling for time across the circadian rhythm. So they're creating these noise variables. And my guess is some of them oscillate with the circadian cycle because they're coming, and some of them don't. Some of them has higher signal to noise and lower signal to noise. So there's a lot of foundational work we need to do to understand these blood biomarkers. But then the question becomes, what's their normal pathology? What's their normal progression? You know, should I see a spike in these in the blood after an injury? And then you get back to normal. And so this might be a route by which we explain a lot of the blood biomarkers for TBI will go up initially like tau and kind of come back down and not be different, even in wildly different severity of TBI as well.
And then just give a shout out to the smartest neurosurgeon I ever met, which was Ed Ofield, who really laid a lot of the groundwork at the NIH for this as a clinician.
I think it's important, you know, when trying to understand places where you've got inputs and outputs that you have to account for, especially if you're looking at biomarkers, you need to know what is going into your system. And so, for example, there's this big issue now in acute brain injury that it doesn't matter how well you improve the cerebral blood flow of a patient. If you measure their oxygen input and their oxygen output, you find that the brain is not actually using any of the oxygen that we're delivering because there's some other problem with diffusivity of oxygen across the blood-brain barrier. Same thing is going to be here. In order for you to interpret your biomarkers, you must know what you're starting with and trying to get out at the same time as you're measuring what that output is.
Also just distribution across the brain, not just across the blood-brain barrier. Egress and the sites of egress are going to be how long and how distributed it is within the brain. And I don't think
Moving a little bit away from cranial nerves, your mention about the neuronal control of the body's lymphatic system.
Yeah.
Common complaint we get in clinic as spine surgeons is I feel my hands are swollen or my legs are swollen and they have cervical stenosis. These are not spinal cord injury patients, so their muscle pumps are working and everything. Do you think there is a correlation between the tightness around the spinal cord or the coda equina and their subjective feeling of swelling?
Absolutely, and I also think this is important in terms of surgical cut downs for various things. We tend to see very large branches, but not most of the stuff innervating the lymphatics is diffuse axons. So we have a special imaging modality that we're using in cadavers to see these things now. And yeah, we're just cutting through that stuff, both from the superior cervical ganglia as well as the vagus beyond the canonical branches. It's shedding these axons non-fascicularly, groups of 20 around the entire length that we're just cutting. And we should have known that because we know you have to get parasympathetic innervation of all that vasculature nearby and stuff like that.
Yeah.
But you got to do what you got to do for desperate subjects, but we need to understand the consequences of that. So it's also an opportunity to maybe we can improve it.
Yeah. Do you think it's there's enough evidence that shows that if you improve lymphatic flow, you will improve the problem with Alzheimer's?
Well, and that's something we're struggling with the biomarkers, right? How much do you need to clear to create a noticeable difference to the subject? Because even in TBI, it's stratified severity, but there's never been, we give them this, we get more of this stuff out. Does it correlate to symptomology? But in rodent models, it appears to.
Cause.
I would be cautioned because physicians have always said something's bad, get completely rid of it, remove all cholesterol. That's happening again. We have a drug to remove all cholesterol. The no cholesterol studies all ended in suicide. It turns out that you need that in the brain. And I think the real thing he's showing here is that this oscillatory motion is a normal motion and that there's a sweet spot. And that homeostasis is really the principle of physiology. What he's telling us is we're going from macrophysiology to microphysiology, and that's fascinating.
We're also trying to avoid augmenting the natural oscillation because they would think there's more likelihood of you doing it wrong in different areas of the brain interfering with it. So we're trying to create a slower oscillatory motion as opposed to trying to respiratory and cardiac gain. The delays in these systems do not allow you to do. There's even changes, the density of sympathetic innervation changes as well. Right to left is different as well. So we need to be very careful we're not manipulating in a bad way.
Thank you, that's amazing. Have you studied deep brain stimulation and the effect of this lymphatics?
Yes, so the rapid circle studies, and we're looking at as well. We've talked to Cam Medical modeling in terms of you're seeing CSF penetration changes about during sleep, and so this gets also interesting in terms of epilepsy and what it sees during night or not. You change the CSF penetration, the field is changing in terms of what you're activating or not. But then there's studies showing that you have to have modeling of these lymphatic pathways at the site of where you're being introduced and things like that as well, and it changes their function. So there's a lot of interesting interactions there. I'm a big believer that people have been thinking they're stimulating continuously at 130 hertz and activating the same volume of tissue. They're not. Every respiratory and cardiac event, you're changing the volume of tissue that you're activating at about a couple 100 mm scale. And then when you sleep, it's changing as well.
We know that sleep improves, it augments lymphatic work. And we know that patients with neurodegenerative disorders have poor sleep. Not focus on improving their sleep as a mechanism for improved lymphatic resonance.
So several people are doing it and looking, and there's multiple studies to try to increase the duration of slow wave sleep. But then the question is, if the system's compromised, can you, if it's not working, does increasing the duration help you? There's also these interesting regulators in terms of if you attempt to do that, that prevents you from increasing it too long as well. So we're hoping that some of the drugs that are being used to increase the duration of slow wave sleep that we can also help with.
Along those lines, maybe we should cut down on the.
Actually, that study said, I looked at melatonin.
Yet still here we are.
All right. So thank you very much, everyone.
Outstanding, thank you.